Very good morning, students. Welcome to biology classes. Today we are going to see 11th science biology, and we are going to see the chapter number 8, that is the cell, the unit of life. And we have already covered so many topics of this chapter, that is so many organelles of the cell, and yet we are going to start with the remaining cell organelles. That is, first we are going to see today is about the plastids, right? So what are plastids? So they are double membrane cell organelle which are found in the plant cells, right? Normally these plastids are found in the plant cells and they do possess double membrane. That means there will be presence of two membranes. For this reason, double membrane cell, it is said to be the double membrane cell organelle. So the plastids are cell organelle, but they do possess double membranes and they are mostly found in the plant cells. They also contain double-stranded DNA and the ribosomes, right? So there will be presence of two things. First will be the double-stranded DNA and second will be the ribosomes, right? So the plastid contain two things, the DNA as well as the ribosomes. Next is they are differentiated into leucoplast, chromoplast and chloroplast. So plastids are of three types, right? So leukocytes, chromoplast and the chloroplast. So we are going to see three of them in detail. First we will see about the chloroplast. So the chloroplast encloses the fluid-filled structure which is known as stroma. So in the chloroplast, right, this chloroplast is being composed of a fluid-like structure in which it is present inside the chloroplast. That means the fluid, it is the organelle which is filled with the fluid, and this fluid part is said to be the stroma. Also there will be presence of stack of sacs which are known as the granum, right? Here you can see in the figure, right, the fluid which is present over here, right, that is the stroma. That is the gray colored, whatever you see at the background, right, that is the stroma, right? And the stack like sacs, right, this are the stack, right, that this is said to be the granum, right? And inside the granum also there will be presence of some fluid like structure, and that is known as lumen, right? Now each of the flattened sac present in the granum is called thylakoid. So here when we are talking about this whole stack, so 1, 2, 3, the each single sac is known as the thylakoids, right? Next is each granum is connected to each other by stroma lamellae. So here you can see this connection between the two granums, right? So this one whole granum has been connected with the another granum. Same way this granum is connected with this granum. So this way the granums are connected with each other, right? So this is what the particular, we can say, that is the stroma lamellae, right? Next, this is all about the chloroplast. Now we are going to see about the chromoplast. The chromoplasts are plastids which are responsible for pigment synthesis and storage. So first chromo, that is, it is imparting or it is producing the pigment, right? So as this particular plastids are responsible for producing or synthesizing the pigments as well as storing the pigments, those are known as the chromoplast, right? They give yellow, orange, red colors to fruits and flowers. So whatever flowers and the fruits, right, you have seen that different fluorescent colors of flowers and fruits are there. So this pigment has been given by the chromoplast, right? Third type of plastids are leucoplasts. So leucoplast as leukocytes, that is WBC, right? So same way leucoplast as this they are colorless plastids. That means they does not possess any of the color. For this reason they are said to be the colorless plastids. And this leucoplast, they act as storage organelles. That means it is doing the functioning of the storage of the organelles, right? Now leucoplast is colorless plastids that are divided into amyloplast that store the starch and elastoplast that stores the lipids in fats and proteins, right? So it is doing, leucoplast is going to store three things, right? Here I have said about that it is colorless plastid, and also it acts as the storage organelle, right? So it acts as storage organelle. That means it is going to store something. So it is going to store three things. First is the starch, second is the lipids, and the third is the proteins. So all the different types of biomolecules are being stored inside the leucoplast, right? So this is about the three types of the plastids. First is the chloroplast, second is the chromoplast, and the third one is the leucoplast, right? So here we complete about the plastids. Next organelle is the most important one, and that is the nucleus, right? In the nucleus, this nucleus is again double membrane structure, right? And this is found in the eukaryotes, right? So in the eukaryotes there will be presence of the nucleus, and around the nucleus there will be presence of double membrane, right? Next is this eukaryotes have single nucleus except the red blood cells, which do not contain the nucleus. Eukaryotes, let's take an example of our own cell, right? So the eukaryotes, that means we do possess, each and every cell does possess the single nucleus, but there will be no presence of the nucleus in the red blood cells. So red blood cells does not possess the nucleus. Next is the nucleus contains the genetic material, which is known as DNA, and this DNA has been packed in the form of chromosomes with the proteins which are known as histones, right? Now this nucleus, inside the nucleus the genetic material is present. That is, genetic material means the DNA, right? So the DNA is present inside the nucleus, and this DNA is not present in the dispersed form, right? This DNA is packed in with each other, or it is packed inside in the form of the chromosomes, right? And it is packed in the form of chromosome with the specialized proteins which are known as the histone proteins. So if it is asked for that, for the packaging of the DNA, which proteins are responsible, or which proteins play major role in packaging the DNA, so those are the histone proteins, right? Next, there are two types of chromosomes which are found, right? So now we are going to see we saw about the nucleus, right? Inside the nucleus there is presence of the genetic material, which is DNA, and it is present in the form of chromosomes. So now we are going to see about the two different types of the chromosomes. So the two different types of chromosomes are euchromatin and heterochromatin, right? Euchromatin is what? It is a less compact structure that can be transcribed, that is formation of messenger RNA from DNA, right? Transcribed, that means transcription is taking place. This you are going to study in detail in 12th standard, right? So euchromatin is what? It is comparatively less compact, right? So it is not that much compact in structure, and here in the euchromatin the formation, the formation of messenger from DNA, it is converting itself to the messenger RNA, right? So that is the euchromatin. And second is the heterochromatin. That means heterochromatin is a compact structure that cannot be transcribed, right? So the one which is not be able to transcribe, that is known as heterochromatin. So euchromatin, that is less compact structure, while heterochromatin, that is more compact structure, right? And euchromatin can be transcribed while the heterochromatin cannot be transcribed. So here the DNA cannot be in the heterochromatin, the DNA cannot be converted into messenger RNA, right? Now the fragment of DNA that codes for a protein is known as a gene. Now there will be presence of several genes in the DNA, right? And this gene is going to code for a particular protein or a specific protein. So the small fragments of DNA which is going to code for a specific protein, that is known as a gene. So there will be presence of the DNA, and on the DNA there will be presence of the genes, right? There will be presence of the nuclear membrane, which is impermeable to the large molecules. So the nuclear pores are present that regulates the movement of solutes in and out of the nucleus, right? Now there will be presence of a nuclear membrane. That means a membrane which is enclosing the nucleus, right? And this nuclear membrane, it is impermeable. Impermeable, that means it does not allow large molecules to enter inside the nucleus, right? So only small molecules only can pass in and out of the nucleus, right? So it allows only specific movement of the solutes only, which is able to pass in as well as out of the nucleus, right? So impermeable, that means it does not allow the large molecules to pass inside or outside of the nucleus, right? So that is the function of the nuclear membrane. Also, there will be presence of a nucleolus, and nucleolus is the largest structure which is found inside the nucleus of the eukaryotes. So in eukaryotes, the largest structure which is found inside the nucleus, and it is what? The nucleolus, right? And this nucleolus is involved in the assembly of the ribosomes, right? So this is involved in the ribosomes, and these ribosomes are none another, they are responsible for the protein synthesis, right? Now, this is the structure of the nucleus. This is nucleus, and there will be presence of this type of pores which are known as the nuclear pores. This is the nuclear envelope, right? This is the chromatin, right? That means the chromosomes which are formed, right, based upon this. So two types of chromatin are there, euchromatin and heterochromatin, right? This, the middle structure which is containing a red colored structure, red colored part, that is the nucleolus, right? Now here in your textbook also it is given about the types of chromosomes, and the first, there are 4 different types of chromosomes which are observed, right? And the 4 different types of chromosomes are based upon the shape and the size of its structure, right? So in the like there are 4 different types of chromosomes, right? And the figure is also been given in your textbook, right? Now, next is the peroxisomes, right? So in the peroxisomes are found in all prokaryotic cells. So these peroxisomes are found in all of the prokaryotic cells, right? Then the major function of the peroxisome is to break down of the fatty acids. So none another, the lipids are being broken down into simpler forms with the use of the peroxisomes. So in the prokaryotic cells, whenever the fatty acids are being produced, or whenever the fatty acids are being present inside the body, the breakdown of those fatty acids is being carried out by the use of the peroxisomes. Now these peroxisomes are derived from the endoplasmic reticulum, right? So now this peroxisome, they are being synthesized, or they are being formed or produced from the endoplasmic reticulum. So the endoplasmic reticulum is responsible for the formation of this peroxisomes, right? Next is the proteins which are found in the peroxisomes, they are known as peroxines, right? So several proteins, right, are found inside the peroxisomes, and these proteins are known as peroxines. As they are produced inside the peroxisomes, the protein name is the peroxines. So this is about the peroxisomes. Next is the cytoskeleton. See, this particular portion is not that much important and is not mostly asked in the exam, but you need to study, because in this chapter, like in the this cytoskeleton, you are going to study in detail in the human physiology. For this reason, here small amount of information is being provided. Now, this cytoskeleton, it consists of a network of proteinaceous filaments in the cytoplasm. So inside the cytoplasm, there will be presence of several proteinaceous filaments which are present. And this is going to form a network-like structures which is known as the cytoskeleton, right? Next is the cytoskeleton provides the mechanical support and aids in cell motility and maintenance of the cell shape. So here what happens, that the cytoskeleton, it is responsible for providing the mechanical support, that is, it is going to provide the support, as well as it is also involved in the cell motility, right? So the cell motility as well as the cell shape, both of them are being maintained by the use or by the functioning of the cytoskeleton. Next is the cilia and flagella. The cilia and flagella, these two of them, they are functioning as functioning for the locomotion of the particular cell, right? So they are hairy outgrowths that function in locomotion. Second, cilia are short and flagella are relatively long. Now the bacteria mostly contain the flagella, right? While we are talking about the paramecium, right, it contains what? The cilia. So cilia are comparatively short outer growths, right? And the flagella is comparatively larger in or longer in size. Now, these two of them, they are covered by a plasma membrane. So there will be presence of a membrane which is known as a plasma membrane around those two, that is the cilia as well as on the flagella. And these two of them, they possess a core which is made up of numerous microtubules which are running parallel to the long axis. So here what happens in the cilia and the flagella, these two of them, they are possessing a core, and this core is made up of so many small microtubules, and this all the microtubules, they are running parallel to the long axis. That means they will be present in the parallel manner, right? So this is all about the cilia and the flagella. Next is the centrosomes and the centrioles. These two of them, they are useful in the cell division part, right? So a pair of cylindrical structures which are known as centrioles compose the centrosome. So when the centrioles are, these centrioles are been arising when the cell is about to divide, right? So these centrioles, they are cylindrical structures. That means the structure of it is of the cylindrical shape, right? As well as this cylindrical structures are present in pair. That means always when a cell is about to divide, from one centriole it is going to separate itself and it is going to form a pair, right? And this pair of cylindrical structures, they are known as centrioles. These two of the centrioles, they compose the centrosome, right? Centrioles are perpendicular to each other, right? Perpendicular to each other, that means they are present on the both of the opposite sides, right? And this centriole is made up of 9 peripheral triplet fibrils of tubulin protein. So this centrioles, they are made up of a specialized protein which is known as tubulin protein. And this tubulin protein, it consists triplet fibrils, right? So there will be presence of triplet fibrils inside it, which are present on the peripheries, right? So this is about the centrioles, right? And the proteinaceous center of the centriole is called hub, right? And it is connected by spokes to the peripheral fibrils. So as I said, there will be presence of the peripheral fibrils, and through this fibrils there will be presence of spoke like structures, and these spokes are attaching the peripheral fibrils to the center part of the centriole, which is known as a hub, right? And the centrioles, which form the base of the flagella and also form the spindle during the cell division, right? So here the centrioles, they form the base of flagella. That means from this region, that is from the centrioles only, the flagella are going to be arise. So that particular region acts as a base for the flagella, and also these are responsible for producing the spindles during the cell division, right? And this you are going to the cell division part, that is the formation of the spindles and everything in detail, you are going to study in the chapter number 10. Okay, now, so this is all about the centrosomes and the centrioles. Now we are going to see the last topic of this chapter. Till here it was about the eukaryotic cell. Now this all the organelles are being observed in the eukaryotic cell. Now we are going to see the structure of a prokaryotic cell. So the prokaryotic cell is represented by bacteria, mycoplasma, cyanobacteria, then PPLOs, and the prokaryotes are much smaller and rapidly dividing than eukaryotes, right? So the prokaryotes, they are rapidly dividing, right? As earlier also in the introductory part I have told you about this. So the particular bacteria, mycoplasma, cyanobacteria, all these things are coming under the prokaryotic cells. Now they have so many different types of shapes, and their organization, that basic organization remains the same. So the way the size as well as the shape is varying. That means the particular shape of the cell as well as the size of the cell, these two of them, they it changes according to the species, but the basic organization of that particular cell remains the same. Now, except the mycoplasmas, a cell wall surrounds the cell. So, except mycoplasma, there will be presence of a cell wall in all the prokaryotic cell. So, in if it is asked that which is the prokaryotic cell in which the cell wall is not present, so that is the mycoplasma, right? Now, this prokaryotic cell, they do not have a well-defined nucleus, right? That means they does not possess a nucleus which is properly properly matured, or there it is not a well defined type, right? Additionally, they also may have some extra nuclear circular DNA which are known as plasmids. It is mostly observed in the bacteria. Now I have also told you about the circular DNA, right? They are said to be the plasmids, right? And the simple short definition of plasmid is what? That they are the extra chromosomal DNA. So none another, the bacteria is already containing its own DNA, but along with that, A circular DNA is also been observed inside the bacteria, and this circular DNA, they have the ability to self-replicate. That means they have the ability to produce its own self and duplicates its own self inside the bacteria, right? So this prokaryotic cell, and mostly in the bacteria, this particular feature is been observed, right? Now, the prokaryotic cell lack all cell organelles except the ribosome. So there will be presence of ribosomes, but except the ribosomes, other cell organelles are absent in the prokaryotic cell. Next is the bacterial cells. If they are motile in nature, so if they are motile, then they may possess one or more flagella, right? So there may be presence of one flagella, right? Or there are possibilities based upon different types of bacteria, right? They may also have more number of flagella, so, right? So if the flagella is present, then the bacterial cell, they are said to be the motile in nature, right? Next, a flagellum has three parts, right? So now this flagellum, it is containing three parts: the filament, hook, and the basal body. Pili and fimbriae help the bacteria to attach to the substrate or the host. So these two are responsible for attaching to the particular substratum, or we can simply say that the attachment is being provided by the pili and the fimbriae. And there will be presence of a pilus, which is help like this pillars can help in the conjugation. That means the sexual contact which is being produced in between the two different cells, right? So this is about the structure of a prokaryotic cell. I hope you are very clear with this chapter. Here we complete this chapter. If you have any doubts related to this chapter, then you can check the YouTube channel for it. I have already prepared the playlist and the YouTube. So if you have any doubts, you can simply check out all my videos which are kept in a proper manner, right? I hope you are very clear with today's topic, and I hope to see you again. Thank you.